So thank you for having me. I'm assuming that everybody can hear me or something like that. Okay, nods, yes, good. And I know that everybody here is looking forward to the idea of listening to a statistician at the end of the day, because that's clearly what you want to do every day to wake you up. But I am going to be talking to you about something that I think may sound sort of counterintuitive, which is the importance of reproducibility. Shouldn't this be presumed? But particularly in the context of high-throughput biology. And the reason I have come to be aware of this as an issue is that I've been working at MD Anderson in bioinformatics for about 18 years. I've seen a whole bunch of people get really, really excited about wanting to take data from new assays like microarrays or mass spectrometry or whatever and determine patient care or be able to treat our patients better. That's the goal. But we have a problem. This is wholly aside from the fact that I have no financial stake in this, that's a different problem. If people want to give me a financial stake in it, talk with me afterwards, we'll deal. But the problem is that once we start looking at measures not of one thing, not of two, but of hundreds, our intuition about what patterns of hundreds of things do, our intuition about what makes sense, it sucks. We have no earthly clue what's going on. And I know this because accidentally, on a few occasions, we have given some of our collaborators completely random lists of genes and still had them construct a story about why exactly those should be changing. So, given that I don't trust our intuition, given that I am suspicious of this, I am faced with a conundrum, which is that we still want to use these high-dimensional numbers to predict patient care. So if I can't trust our intuition, that means that we've got to be able to trust the process through which we assemble these, and we've got to be able to check those individual steps. Okay, we can deal. But then we face another challenge, which is occasionally we go to the literature and we are inspired. We see Nature papers saying, wow, this is wonderful, we can treat our patients better. I know, before we try it here at MD Anderson, we'll just read through the paper, read through their methods, grab the data, and make sure that we can get the numbers they report. Surely that's a trivial exercise because they've posted all the numbers that they have and all their data and things like that. Has anybody here tried this particular experiment? Those of you who have will recognize that this statement is an ideal, not an observed fact. But this faces us with yet another challenge, which is that on occasion, the papers are so compelling and our collaborators are so invested in wanting to get it to work that we are forced to do something which is involving an entirely new branch of bioinformatics, which is forensic bioinformatics. And what this is, it is the art of saying, here's the data they started with, here's the results they reported, what the hell must they have done to get from here to here? And this is occasionally informative, and it is turned out to be informative about aspects of high-dimensional biology and basic statistics in a few unexpected ways. So to illustrate this, I want to walk you through some case studies which are important in the diagnosis and treatment of cancer. So the first case study I'm going to talk about is something that some of you may have heard of. This was an example that started a few years ago where investigators at the NCI and the FDA, as it happens, were very excited, and they fast-tracked a paper in The Lancet where they said, we've figured out how to use mass spectrometry profiles of serum samples to diagnose whether or not a woman has ovarian cancer. Now, this would be a good deal from a diagnostic perspective, because we still don't have a very good non-invasive test for ovarian cancer, which means, of course, that by the time we actually see the patients with it, it's had time to grow because the symptoms are not really all that specific. They're things like bloating, constipation, etc. My wife's a doctor. I had her describe the list to me, and I said, those aren't very specific. And she said, no, those symptoms occur with all my patients, including the men. So it's probably not specific to ovarian cancer. Okay, fine. So what did they do here? Well, they took serum samples from 100 women with ovarian cancer, serum samples from 100 women who were healthy controls, and 16 from women with ovarian cysts, relatively benign disease, painful, not going to kill them. And they said, what are we going to do? We're going to take our patterns, our mass spec traces, and we're going to take half of the cancer traces and half of the control traces, and we're going to toss them into a wonderfully complex machine learning algorithm, and we're going to come up with a classifier, and then we're going to drop each of the spectra we haven't seen yet into this classifier and see how well it does. And the reason I'm talking about this is that their results, when they dropped the new spectra into their classifier, were pretty darn good. They said, oh, every single one of the cancer cases we got right. We said this is a cancer. They didn't get all the normals right. They got confused on a few, but they still got most of them right. But even more, for the 16 samples from women with ovarian cysts, when they dropped them into their machine said, this is not cancer, but it's not normal either. Now that's interesting because it suggests that the pattern that they are exploiting is specific to ovarian cancer, not general disease. That's actually pretty cool. So this meant that whole bunches of people at MD Anderson got excited. 8 different labs bought separate mass spec instruments and started sending their data to us. They spent a whole bunch of millions of dollars setting up a separate proteomics facility, and they all started coming to us with data saying, we want to use this. And we said, well, great, we'd love to help. Maybe we can help by looking at the data and figuring out how we can apply this here. And the thing about this, and one of the nice things about this particular study, was that the initial investigators posted their data to the web, okay? And actually, by the time we got to looking at it, they had posted not just one data set, but actually 3. So let me tell you about these 3 data sets. The first data set that they posted was a bunch of spectra. It's 216 samples. These are the spectra that served as the basis for the claims in their Lancet publication, okay? 100 spectra from cancer patients, 100 from healthy controls, 16 from women with benign disease. And the spectra that they posted, all of the samples, all the spectra had been baseline subtracted. I'll show you what that means in a moment. And all of them had been run on a chip using a particular surface chemistry that's called an H4 chip. And they said, we got great results. The fun thing here is that the company that was producing the chips that they were using, the physical surfaces, said, actually, if you want to look at serum, that probably isn't the best surface chemistry to use. You should use this other one. So the group in question said, all right, we'll take our samples, we'll run it using the chip you recommend. It's a different surface chemistry, same samples, and they reported great results again. Data's posted, baseline subtracted, fine. And then they said the question is clinically important enough that we want to verify it. So we're going to take some new samples, new women, some with ovarian cancer, some healthy controls, no benign disease this time, and we'll run it on the WCX2 chip, that's the better one for this type, and we're not going to subtract the baseline this time, and they reported great results again. 3 data sets, 3 great results, wonderful. And the fun thing here is that every time you run these mass spec things, every trace has 15,000 observations in it. But they also said that if you want to separate our cancers from our controls, we're going to tell you on a case-by-case basis that you don't need all 15,000. You need somewhere between 5 and 7, and we'll tell you, for this data set, here's what they are. For this data set, here's what they are. So, okay, sounds cool. So we've got the data. We know what peaks they use. We downloaded the data and said, can we replicate their results? Can we achieve the separation between cancers and controls they are discussing? And the answer is no. The answer is no for those first 2 data sets because the data they posted is qualitatively different from the data they used to build their classifier. The data that they used to actually build their classifier was data as it came off the machine. This is raw spectra. You will notice that there's this low frequency hump, etc. That's because with the machine they're using to get the proteins loose from a matrix, what they're doing is they're blasting it with a laser. When you blast stuff with a laser, if you're going to blast it hard enough to kick heavy proteins loose, it turns out that that's hard enough to kick up a crowd of, well, scientifically described crud at very low masses. So this down here, if you're going to measure up to about 20,000 Daltons, which they were trying to do, that means that stuff below about 1,000 is probably garbage. And normally, what you don't want to focus on this low frequency trend. You want to focus upon where the peaks are after you get rid of the low frequency trend. But they found the patterns here. When they posted this, the signal that they had found went away. That's a bad sign. So we looked at that and said, well, that's disturbing. Can we still do something? I mean, we've still got the spectra. They've been baseline subtracted, and actually now at this point, we've got the spectra we really want to use. Can we see some type of separation that we can exploit? So we said, let's take a look at the spectra from the first data set that they looked at. These are the 216 samples. 100 from cancers, 100 from healthy controls, 16 from benign disease. The bright colors, the bright yellow, those are the locations of the intense peaks. The x-axis is time of flight, the y-axis is sample, okay? But I have a deep statistical question for the audience here. See, this is even better. You're gonna have not just a statistician, you're gonna have one who's asking you questions. Question here, which of these three groups is not like the other two? It's the others, which the thing is, that's really weird because as we understand the biology, cancer is a big time screw up of cells, not ovarian cysts. So we expected the division between cancers and normals, not down there. So we said, what can we do? We know, we're academics. We'll write a paper about ovarian cysts. So we said, okay, great. And even better, because they run these same samples again, we should be able to learn even more about the proteins involved. So let's take a look at the same type of heat map for the second data set. And then we stared at that and we said, well, that's unexpected. And then we also tried something further, which is something I have learned to do as a bioinformatician over a few years, which is every once in a while be just a bit paranoid about your data, which means we plotted both data sets at the same time. And then we looked at the location of the most intense peak down here, and we drew our eyes upwards. They shifted the chip type before they were done with the first experiment. Can they tell the others from the other two groups? Yes, they can. Is it biologically relevant? No, it's not. Furthermore, what does this tell you about whether or not they randomized the order in which the samples of the first experiment were run? I'll tell you that if they did randomize it, we've got a p-value of 10 to the minus 200 going on. So no. What we have here is an issue of complete confounding of the experimental design. A big change is associated with a change in measurement. Okay, fine. Well, that means that we're not too happy with the first few data sets. We've got this baseline subtraction, which kills things. But wait, to quote Yoda, there is another. There is the third data set, which they didn't subtract the baseline, so we should be able to exactly get what they did. So, okay, for the third data set, they said, here's the 7 peaks that we used. So we took, for each spectra, a 7 vector, and we computed pairwise distances between samples, and that's what I'm plotting here. And what this plot shows is something we want to see. In the upper left, we're seeing distances between pairs of cancer spectra. Cancer spectra are close to each other. The distances are small. Healthy spectra, control spectra, those are also close to each other. The distances are small. The distances between cancers and controls are big. We have good separation of the two data sets. Cool, wonderful. But, different thing. They said that they used 7 peaks to accomplish this. Now, the 7 peaks that they used, they identified using their super spiffy machine learning algorithm. We didn't really understand their super spiffy machine learning algorithm. We're simple folks. So we said, is there something else that we might try? Well, there's about 15,000 observations, so actually what we did is observation by observation, value by value, we compared the cancers and controls using 2 sample t-tests. And then we said, some of these t-values are pretty darn big. I'm used in real data to seeing a t-value of maybe 4 or 5 if it's exciting. We have t-values in excess of 20. So we looked at the 7 peaks that they said were important. And of the 7 that they said were important, 1 has a t-statistic value bigger than 20. A second has a t-statistic value bigger than 10. And the other 5 all have values that are very, very small. Maybe we don't need the other 5. Maybe it's easy. So we decided to take a look at what we could do just with the 2 best. Guess what? We can achieve perfect separation with 2 peaks. That's all we need. But let's take a look at the masses involved here. They're below 500. What did I say was the cutoff for garbage? So we're not too happy about this, but still this does suggest a strategy. We identified 2 peaks because they had really big t-statistic values. Okay, we've got this entire vector. What if we just take a look at every pair of peaks that has a t-statistic or 10 or more? Can we find another pair that separates the data? Because then maybe we can use that. So could we find another pair that perfectly separates the data? Yes, we can. The masses, however, 245, 2.8. cancers and controls differ in their ability to incorporate different isotopes of hydrogen. That's not how it works. Actually, the thing is that 2.8, that is not real. That is because the machine's calibration does not extend down that far. And actually, if you look really closely at the raw data, that is from a part of the spectra before anything could have physically reached the detector. We are seeing perfect separation of the samples being driven by electronic noise. That happens because, as it happens, they ran all the cancer samples a few months after they ran the control samples. We again have perfect confounding of the experimental design. Now, while all this was going on, the investigators in question got together with a separate company, and they went in 2004 to the annual meeting of the Society for Gynecologic Oncologists and said, we are going to, in a few months, be offering a particular diagnostic test. You send us a serum sample, we will tell you if this woman needs her ovaries removed. We were not happy. We wrote up some of the objections we had. We sent our objections first to The Lancet, which is where the initial results had appeared. The Lancet told us that the topic we were raising, which was complete confounding of the experimental design, was too technical for their readership, so they weren't going to publish it. So we said, well, that's rather disturbing, so we decided to send it to bioinformatics, because most of the MD community reads that too, so that's okay. And as it happened, our paper came online at the end of January, and the next week, the New York Times covered the story because they didn't think the issue was too technical for their readership. And a few weeks after that, the Society for Gynecologic Oncologists issued a position statement saying, we don't think this is quite ready for prime time, and the FDA started sending polite letters to the companies involved saying, would you please hold off on advertising this until we have a chance to talk with you? And it is at this point, and over the next few years, that the FDA decided that it was going to assert that when we are using complex signatures, multi-omic signatures, which we don't fully understand, to determine medical therapy. These are in vitro diagnostic multivariate index assays, IVD-MIAs. In the eyes of the FDA, those are medical devices and subject to regulation as such. And in this particular case, they said, you may not market this until you've proved to us that it works. It's still not on the market today. Now, okay, that's one thing, but surely, I mean, this story is old. Surely we've gotten better. I mean, this is the story from 2004, and I post it here because I'm vain. But there was another story that ran in the New York Times 4 years later, which shows that the editors at the New York Times really need to be more imaginative. And this was about another proteomic study, also involving ovarian cancer, and it turns out that there were indeed some proteins that were different between the two groups of women. What's going on? Well, it turns out that the serum samples from the healthy controls were collected from women at the time of their annual gynecologic exam. The serum samples from the women with ovarian cancer were collected from them before they were wheeled into surgery. The peaks were associated with stress proteins, and it turns out that women about to undergo surgery for ovarian cancer are more stressed than women who are in for their annual gynecologic exam. Who'da thunk? So this is probably not completely fixed here. Now, who here thinks that this issue is unique to proteomics? Good, okay. So we took a look at this in the context of microarray data a few years later, and this is one where they were trying to predict response to different therapies in a set of cohorts of patients. You see these nice 3 blocks here? These 3 blocks in the microarray data, those actually correspond to 3 qualitatively distinct run dates for the assays. This 3rd block right here, the reason this is red, that's all of the patients from one of the treatment arms. Again, we have complete confounding of the experimental design. As it happens, all of these assays were not only run several months later than everybody else, they were run in a different lab in a different country. So we've got some big differences here, and experimental design is important. Now, that is something that we can pick up from the data, and that's useful. But since I've shifted arrays, I also want to shift to a different example, which is to talk not just about diagnosis, I also want to talk about treatment. So about this time, as we were looking at this, another paper appeared where a group, and this showed up in Nature Medicine, where a group at Duke claimed to have solved another problem that's clinically very important. Specifically, for cancer patients, they would be able to say, which drugs will work for this patient. We know this is important because if a woman shows up at MD Anderson with ovarian cancer, we will give her the standard frontline therapy, which is a platinum agent and a taxane. And we will do this knowing that it's going to help roughly 75–80% of the women we give it to. It's going to improve their quality of life. It's going to make them live a bit longer. But for the other 20%, we're going to poison them for 6 weeks to no benefit. But we're going to do it because we do not know going in which 20% they are. If we did know which 20% would not respond, we'd do something different. And that is the problem they claim to have solved. How'd they do it? Well, they said, we're going to do this by exploiting the potential of a brand new assay that is available to us. And at the time, these were microarrays. Brand new, okay. So here's how it works. We're going to start by looking at not patient samples initially, but rather data from cell lines. And it's a very special panel of cell lines. It's a panel known as the NCI-60. This is a panel of cancer cell lines that has been maintained by the National Cancer Institute, the NCI, for about the past 40 years. Why has the NCI gone to the trouble of maintaining this? Well, one reason is that in order to figure out the mechanism of action of many of the chemotherapeutics drugs, one of the ways that the NCI does this is every time a new drug is introduced, they will acquire some of it and they will assay it for effectiveness against every cell line in this panel. They will say, for this drug, what concentration of the drug is required to cut the growth rate of this cell line in half? And the fun thing is that this data about drug responsiveness to in these various cell lines, all of this is public. You can download it for every chemotherapeutic now in use. So, if there is some way that we can generalize from the sensitivity profiles of cell lines to the likely sensitivity of patients, we have in this particular data set something that in essence might serve as a Rosetta Stone for saying what drugs would work for what patients. So here's how they exploited it. They said, let's take a drug of interest. Let's say it's Docetaxel. We're going to take all of the cell lines in the NCI-60. Quick quiz for the audience. How many cell lines are in the NCI-60? Everybody got 59, right? It pays to look at the data. The fun thing here is that they're going to take, let's take the most sensitive and most resistant, let's say 7 at each end, and then we're going to look at microarray profiles of these samples. The microarray profiles are also publicly available data. The microarray profiles have measurements on about 20,000 genes. We're going to compare the genes across groups using immensely sophisticated mathematics like two-sample t-tests. And having done this gene by gene, we will take the top 50 or so and we will say the pattern of expression in those top 50, that comprises the signature of likely sensitivity to Docetaxel. We will then look at a microarray profile of a patient, look just at the values for those 50 genes, and we will say, does the pattern for those 50 looks more like the sensitive cohort or more like the resistant cohort? If it's more like the sensitive cohort, administer Docetaxel. If it's over here, give them something else. And the reason this got into Nature Medicine is that they said, using publicly available data, we were able to test this and its predictive ability for 7 commonly used drugs, and we got great predictive results every single time. Which meant that within weeks at MD Anderson, we had a bunch of groups at MD Anderson coming to us in bioinformatics saying, we want to do this. We want to improve patient care at our institution. We want to use this method, but this method involves math, which is scary. So, we want you to help. And we said we would be delighted to do so, and we would be delighted to do so for a few different reasons, but one of them is that all of the data that they're using is public. So we should be able to check it at each and every step. So we said, let's begin with checking a step where we think the answer should be fairly easy to check. In particular, let's pick one of the drugs they looked at, and let's see if we can match or figure out if our selection of genes that are important matches theirs. Why did we think this would be easy? Because one of the supplementary reports associated with this paper says, for this drug, here are the genes we found. For this drug, here are the genes we found. And within the paper itself, for several of the drugs, they said, this obviously makes sense because these genes do this. And so there was an explanation going on. So we said, all right, let's take one of the drugs, 5-fluorouracil. They reported identifying 45 really different genes. Okay, fine. We got the arrays. We did the 2-sample t-tests ourselves. I have a PhD in statistics. I can do this. and we got R45, and we looked at R45 and their 45, and the intersection between these two sets was 0. And we said, that's probably not good. What's going on? Well, as it happens, when you start measuring not one gene, not two, but thousands, you often start losing track. So you don't keep track of them by name anymore. You sort of keep track of them by some type of abstract index. And in this case, the arrays in question were manufactured by Affymetrix. So the natural identifier was an Affymetrix probe set ID. Why does this matter? Well, let me show you. On the left, we have the first 4 Affy probe set IDs of genes that they identified as important. On the right, we have the first 4 Affy probe set IDs that we identified by 2-sample t-tests. As you go from the list on the left to the one on the right, does anybody see a pattern here? We have an off by 1 indexing error. What's going on? Turns out that we tracked down the software that they used. The software that they used required 2 inputs. The first is a big gene expression matrix, genes by samples, with a header row that says, is the sample sensitive or resistant to treatment? The second input is a list of gene names, same order as the matrix, but for their software to work correctly, this list of gene names must not have a header row. So if you're working in Excel, and you cut, and you paste, and you leave on that header row, guess what happens? You get an off by 1 indexing error. Now, what does it tell you about the fact that they were able to explain why this list of genes made sense when none of them actually fall out of the mathematics? Our intuition in very high dimensions sucks. And this problem is present for every one of the 7 drugs they looked at. It got by the authors, it got by the reviewers, it got by the editors, because we're not equipped to pick up problems like that. But as it happens, this problem is embarrassing, yes, but it's actually not fatal. It's not fatal to the clinical goal that we want to do or want to achieve, which is what we're actually trying to do is we're trying to use the gene expression values, the numbers, to predict patient response. They're finding the predictive patterns and affixing the gene names at the end so they can explain it, but that's the last step. If the numbers are still capable of predicting patient response, we can still use this. So we said, all right, we're going to try that. Can we make predictions using these numbers that are still pretty good? So how are we going to define pretty good, or at least good enough? We decided to say we understand this well enough to use it if we can get predictions as good as they got. They got it into Nature Medicine. Those must not be too bad. But it does raise the next question of how good were their predictions? Well, here's an example. In the top panel, which is taken from their Nature Medicine paper, these show the predictive scores for women with breast cancer who were treated with single-agent docetaxel. They predicted that most of the women who are actually sensitive to treatment, blue squares, turns out that most of the sensitive scores were low. That's pretty good. Most of the resistant scores, red triangles, they were high, pretty good. So there's pretty good separation, and if you set the cut point around the middle, it turns out that you correctly classified 22 out of 24 samples. That's the rate they were claiming. That's not perfect, but it's not bad. But as it happens, this also raised a slightly different question for us. So I have another statistical analysis question for the audience. Specifically, if we look at the sensitive samples, the blue squares, how many blue squares are there in this plot? It's close to 12, not quite. 13, there we go. We have 13 blue squares and 11 red triangles. Minor problem. I told you that all the data was publicly available, including the stuff for the data sets they were using for validation. Well, the initial data set came from a paper in The Lancet 3 years before, where the authors of that paper spent a lot of time talking about their 11 sensitive and 13 resistant patients, not 13 and 11, which we thought might be a problem. So we decided to take a look at the results for a different drug. For those of you who are feeling overly ambitious, how many red triangles are there? Okay. Main thing here is that, okay, again, they're claiming good overall accuracy for treatment with a drug to which most of the patient population is resistant. The problem here. The source paper for this data set is a paper from the New England Journal, which is about the treatment of childhood ALL, a form of leukemia. The thing is, as cancers go, this is one we know how to treat. We have cure rates of 80% or so. What does this mean? It means that if in this patient population we have a drug to which most of the patients are resistant, we wouldn't be using it. And actually, the authors of the New England Journal paper thought that most of their patients were sensitive to treatment, not resistant. What's going on? Well, we went back to the software they were using. I told you that that first input was this big expression matrix with a header row with sensitive and resistant at the top. Well, strictly speaking, what that header row consisted of was zeros or ones, which they were using as coding. But they never explicitly wrote down 0 equals sensitive, 1 equals resistant. So in some cases, going from one drug to the next, they got the labeling reversed. So what would this mean clinically? It would mean that if you were to use these rules to decide on patient therapy, you would recommend administering Adriamycin just to the patients who were most resistant to it. We think this is poor clinical practice. We do not recommend this. So now, by the way, this reconstruction that we have just discussed, this is not something that took us 5 minutes, 10 minutes. No, this actually was the result of about 6 months of email interactions between our group and the group at Duke saying, we would love to use this method. Can you help us? By the way, some of the labels appear weird. Maybe we can fix that. By the way, some of these other labels appear weird. At the end of this period, we had to tell them that actually, when we use the labels we think are correct, we don't think your predictions work. And they said, well, obviously this interaction has ceased to be productive, so we want to bring it to a close. And we said, okay. So we took our various objections and we wrote it up and we sent it as a letter to Nature Medicine, where it appeared the following November. And we were waiting to see it appear, amongst other things, because we were curious as to what the author's response would be. I mean, we were critiquing things, they have a right of response, and we weren't sure if they were going to say, oh, we screwed up, we're so sorry, we'll never do it again. But that, as it happens, was not the tone of the response. The response was that the analyses by Drs. Baggerly and Coombs are deeply flawed. They have not followed our methods. And actually, no, our labels for our samples are correct, including for Adriamycin with all the red triangles. No, our labels of sensitivity are correct. We've posted data to our website, so we've got that right. Furthermore, we have continued to apply these methods in other high-profile studies and gotten great results again with editorials and scientific acclaim, so obviously we must be right and they must be wrong. As it happens, this came out on the evening of November the 6th of 2007. I remember this because I was in a hotel room at Bethesda, Maryland, because I was supposed to give a talk at the NCI about this topic the very next day. So right before I was about to give this talk, I had just been told that everything I was going to be talking about was wrong. This made me slightly disgruntled. So I said, well, all right, they say that we've gotten the labels wrong. They say they've posted the data. Well, let's look at the data. Maybe we screwed up. Downloaded the data for Adriamycin. This is a big Excel table, 144 columns, about a few thousand rows. The rows are genes, columns are samples, and there are 2 header rows. The 2 header rows say, are these samples cell lines or patient samples? Okay, fine. And they say, are the samples sensitive or resistant to treatment? Count the number of patient samples who are resistant, number of red triangles, matches their number of red triangles. Okay, fine. Well, maybe they found something. So we decided to maybe cluster the data. So actually, to do that, we actually looked at all the pairs of samples and we looked at the correlations between pairs. And I've plotted, in this case, for all 144 samples, where the correlations are really high. In the lower left, these are the cell lines. This line of red dots on the main diagonal, what is this saying? This is saying that cell lines are highly correlated with themselves. I believe that. That's good. Okay? Up here are the patient samples. And this line on the main diagonal we expect. These we don't. What's going on? Well, if you look more closely at the data, while they have 122 columns that are labeled as patient samples, those columns of numerical values are not all distinct. Some columns of values have been reused 2, 3, or even 4 times. So we don't have 122 distinct patient values here. Even better, right about here is data from one patient whose numerical values were used 4 times, where that one patient was labeled resistant 3 times out of 4. So we're pretty confident the labels aren't right yet. So we wrote this up and sent this as a note to Nature Medicine. Nature Medicine said, oh, the authors will make a correction. There's no need to publish your objection. So we said, okay, fine. And the next year, the authors did publish a correction where we said we redid the analysis using only the 95 unique samples. We got great results again, so great. Minor problem was that we'd only counted 84 unique samples, so 95 was a surprise to us. We looked at the 95 they now listed. Of the 95 they now list, there's only 80 that are distinct, 15 are present twice. Of the 15 that are present twice, 6 are labeled both sensitive and resistant. So the correction of a correction still needs correcting. But Nature Medicine had said it was done, so we moved on. We moved on instead to look at some of the papers they cited as providing further validation of their approach. And so here's one of them. This showed up in the Journal of Clinical Oncology, and it extended their approach to dealing with 2 other drugs, specifically cisplatin and pemetrexed, commonly used in the treatment of lung cancer. And they said, these signatures look persuasive to us, in particular because the signature for cisplatin includes genes such as ERCC1, ERCC4, and other DNA repair genes. And that's persuasive to us because there have just been a few papers in the New England Journal saying cisplatin acts in part through this pathway involving these genes. So okay, so they said it looks persuasive to us. The lung cancer group came to us and said, could you check this? Could you tell us if we should use this? So we went through and we looked at their lists of genes for the 2 drugs. And for pemetrexed, they report 80 genes. With some work, we are able to match all 80 of the genes that they identify, after we allow for the off by 1 error, which they are still making. For cisplatin, they report 45 genes. With some work, we are able to perfectly match 41 of the 45 genes, after accounting for the off by 1 error. 4 of the genes they report do not fall out of their software. What are the 4 that their software does not produce? One is a probe set for ERCC1, another is a probe set for ERCC4, a second probe set for ERCC1, and another for a DNA repair gene. These are the ones that establish plausibility. Well, that's interesting. But the thing is, even in this context, of these 4, the bottom 2, those bottom 2 are special. What makes those 2 probe sets special? What makes those 2 probe sets special is that those were not physically present on the assays they used. They were actually using arrays that were part of a 2-chip set covering the genome, the A and the B. The A contains most of the named genes, so to economize, that's the only chip they ran. 2 of their best genes haven't been measured yet. We thought this was a neat trick. We sent a note about this off to JCO. JCO sent us a note back saying, we're sorry, we can't publish this. We sent them a second note saying, could you tell us why? JCO sent us a second note saying, we're sorry, we can't publish this. So we moved on. And over the next few years, we moved on to look at other papers from this group and to be repeatedly told that, well, no, we don't need to publish this objection. My favorite was from the Lancet Oncology, which said we were arguing about statistical issues to which everyone knows there are no right or wrong answers. So we decided not to worry about it. Now, there is this issue of, okay, we're going on with this. Why do we care? Why we care was that about 2.5 years into this, we learned that the group at Duke had already started clinical trials where they were using these rules to allocate patients to treatment arms. In some of these trials, based on the data they had posted, these were using drugs for which they had the sensitive and resistant labels reversed. We weren't happy. So we wrote up our various objections in the form of a paper. First thing we did was we sent it to the editor of a prominent biological journal for informal comment. His comment to us was that your story is too negative. Can you fix that? And we said, you know what? We really don't think we can. So the next thing we did was we sent it to the Annals of Applied Statistics. Again, because we thought most MDs read that one too, right? Okay. The response there was a bit different. I got a note from the editor that evening saying, you know, I like this paper. I'm going to handle its review myself rather than handing it off to an AE, which I took as a good sign. And I took as a better sign the email I got 2 weeks later where he said, we've simply decided to accept the paper as is. It's posted online now, which means I'm sending more papers there. So that's a good thing. But the thing is, the paper is now out. It's now out in the published literature, which means we can cite it as we perhaps contact members of the press. So we had a nice discussion with Paul Goldberg of the Cancer Letter, and indeed Paul Goldberg, a few weeks later, wrote a story in the Cancer Letter saying, biostatisticians at MD Anderson are alleging the potential for patient harm in clinical trials being run at Duke University. Oh, the horror! And unbeknownst to us, some folks at the NCI read our paper, and after reading our paper, looked at grant applications from the same group. And they said, we see the same mistakes here. So the NCI wrote a note to Duke's IRB, their Institutional Review Board, the group in charge of patient safety, saying, we are concerned. Can you reassure us? Now, I'm a professor at a different institution. I can yell all I want. There's a limited amount that they're going to do to appease me. The NCI is funding Duke to the tune of tens of millions of dollars a year. Duke pays attention. So the following week, Duke said that, okay, we're going to suspend enrollment in these clinical trials, and we're going to convene a panel of experts to examine the science and see whether it's justified and whether we should continue. And we breathed a sigh of relief. We said, we're really upset that it got this far in the first place. But given that it did, we can accept people just looking at the science and clarifying it. That's fine. We'll wait and we'll see what happens. And we said, all right, well, while we waited, the first thing that happened was that the group at Duke posted brand new data to the web. What was the new data? It was data that they said, this is the validation data that we use to prove that our signatures for cisplatin and pemetrexed work. Okay, well, where did that validation come from? Well, the validation data is 59 samples from a larger data set of 153, and this larger data set of 153 is publicly available. So we said, great. We downloaded all 53. We downloaded their numbers. We checked them against each other. We checked the correlations. We plotted the data. And if the samples are what they say they are, what you should be seeing in this plot are 59 red squares all on top of this blue line. How many red squares are on top of that blue line? Try again. 0. As it happens, in this part of the thing is green and red are a bit confused on this particular display. But as it happens, in this plot, there's only 43 red squares at all. For those 43 samples, when we look at the numbers, we can perfectly match the numbers to the source material, and they got the name wrong. It's not the sample they say it is, which means they got the clinical annotation wrong. For the other 16 samples, there's no red squares. Why not? Because for those samples, they've already scrambled the gene names so badly that this is no longer a real sample of anything anymore. But what we can tell you is that the data for all 59 validation samples is wrong 2 years after they started clinical trials with these drugs. We wrote this up as a formal report, which we sent to Duke and to the NCI. And we waited to see what would happen next. And what happened next is that Duke announced that they were getting ready to restart the clinical trials, because they had finished their scientific investigation, and their investigation's results had strengthened their confidence in this evolving approach to personalized cancer treatment. This surprised us, and we said, would you show us the data upon which this decision is based? Duke said no. Duke said, while the reviewers approved of our sharing the report with the NCI, we consider it a confidential document. A future paper will explain the methods we are using. We will treat the patients this way now, and we'll explain why it makes sense later. Well, we stared at this for a while, and then it actually occurred to us that this may have given us something else to try. Let's take a look at what Duke actually said. While the reviewers approved of our sharing the report with the NCI, and let's stop there. The NCI, the National Cancer Institute, is a federal agency, which means it is subject to the provisions of the Freedom of Information Act. So, that May, we obtained a copy of the report from the NCI pursuant to a FOIA request. And we looked at the report together with a bunch of other statisticians, and we said, this does not justify restarting trials. What the hell's going on? And Duke had no comment. And we were sort of depressed because we didn't really see what else we could do. And I was sitting in my office an evening after this, and I got a phone call. And the guy on the phone said, hi, I'm a lawyer. My client wishes you to be aware of some of the following things going on in these particular clinical trials and involving the PIs. I had a nice chat with him, and I said, that's really interesting. Here's the phone number of a reporter you should be talking with. And about an hour and a half later, I got a call from Paul Goldberg of the Cancer Letter saying, I'm going to have fun. So why was Paul going to have fun? Well, it turns out that 2 weeks later, Paul got to run a story pointing out that one of the Duke investigators used to claim on his CV and on grant applications that he was a Rhodes Scholar, but the Rhodes Foundation disagreed. We know more about this because he said, well, maybe I should talk with Paul. After all, I was a nominee. And the reason we know this is that Paul Goldberg had emailed him a whole bunch of questions, CCing a bunch of Duke deans as he did so, and Poti typed out a reply, and he hit reply to all. So what has just happened? You've just had a member of your faculty admit in writing to a member of the press while CCing your university's administration that, oh, by the way, I've been committing fraud for the past 5 years. He was placed on academic leave that day. That was Friday. Now, as it happens, a bunch of friends of mine had been talking or getting tired of me yelling about this for a while, so I spammed them with this article. And I said, I know some of you have been talking about writing a letter. Now is politically the best time you are ever going to have. So the following Monday, a letter from a whole bunch of biostatisticians landed on the desk of Harold Varmus, the brand new head of the NCI, saying, would you please resuspend these clinical trials until the science can be clarified by an objective outside party? Varmus called Duke and said, you're going to suspend these trials, aren't you? Duke said, yeah. Varmus called the Institute of Medicine and said, would you act as an impartial third party? The IOM said, yes, it's gotten stinky enough, we'll do it, we'll start in December. Before they got there, however, something else happened. Joseph Nevins, senior author on some of these papers, sent an email to some of his collaborators saying, you know what, we may have to retract one of the papers. What paper were they going to have to retract? The one dealing with cisplatin and pemetrexed. Why were they going to have to retract it? Because on closer examination of the database, it appeared that 43 of their validation samples were mislabeled, and they could not verify the other 16, which is what we had told them the previous November before they enrolled more patients. The next week, Duke officially terminated the clinical trials and announced that in retrospect, these trials should never have been run. A week and a half after that, Poti resigned from Duke, and a cascade of other things began, including lawsuits, a misconduct investigation, a bunch of retractions from such fly-by-night journals as the New England Journal, JCO, JAMA, and the like. It also hit the front page of the New York Times. It also hit 60 Minutes. And I'm going to show you a clip from 60 Minutes, and this is an interview between Scott Pelley, CBS News anchor, and Joseph Nevins, the senior author.
Dr. Nevins discovered that when the underlying data disproved Dr. Poti's theory, the data were changed.
It became clear that there was no explanation other than there was a manipulation of the data, a manipulation of somebody's credentials, and a manipulation of a lot of people's trust.
Manipulated data. These were not errors.
That's correct. It simply couldn't be random. It simply couldn't be inadvertent. It had to have been based on a desire to make something work.
Is it a close call, or is it abundantly clear that the data were fabricated?
Abundantly clear.
That episode aired 5 years after clinical trials began. It's abundantly clear the data were faked, but the data were never double-checked until years after the trial began, and indeed after all this crashed down. Some other observations. This is an aberrant case. I will concede that. Indeed, this is in many ways a case study of exactly how not to do things, and involves a whole bunch of mistakes. But I'll tell you that these mistakes here, and in our earlier example, we recognized them. We were able to detect them. Why were we able to recognize and detect them? Because the types of mistakes we're talking about, confounding in the experimental design, mixing up the sample labels, I've seen each and every one of these mistakes in data sets that have come to me at MD Anderson. This is not because MD Anderson is a bad institution. It's not. It's a very good institution. It's because some of these mistakes are easy mistakes to make, particularly if you're treating Excel as your database. And the thing is, because they're easy mistakes to make, if you supply us with the full data and documentation, they're often easy mistakes to fix. So if we can ask the PIs to, will you send us the rest of the stuff, we can typically fix them pretty quickly. But if you take something, you make an easy mistake, and then you say, well, the journal won't give me space to post all the supplementary material I want. I know, I'll just give them the data set and not anything else. It's now a lot harder to fix. Now, I have just told you about a pathological case. Should we be worried about a pathological case? The problem is the issue is not restricted to one paper. How big is the problem of reproducibility? Perhaps the thing that got the NIH's attention the most was a survey paper in 2012 in Nature by Glenn Begley and Lee Ellis. Glenn Begley headed clinical development at Amgen, and in this paper they said, we want to tell you about our experience at Amgen where working with the authors of 53 breakthrough papers in our subjective assessment, we tried to replicate the results so we could commercialize them. Working with the authors, we were able to replicate the results in 6 of those 53 studies. That scared the NIH, because they say, we're funding most of these studies. We don't want to go before Congress and say our validation rate is just a bit above 10%. So we want to do something better. And this has led to various things, including, as of 2016, a rigor and reproducibility initiative launched by the NIH to do things better. Now, why are they doing this? Here's some of the dollar figures involved. So their research budget is about $56 billion a year. Roughly half of that, by the various estimates, is probably wasted. How does that break down? Biological reagents and reference materials, this is, did you get the labels right? Study design, this is, did you avoid confounding? Data analysis and reporting, this is, did you tell us what you did? 90% of $28 billion lost due to mistakes we can easily avoid if we're aware of them. That's useful. Now, we actually were asked what we would suggest. We said that actually, for high-throughput studies in particular, we've reached the point where if you're going to publish, we need the data and we need the code because written descriptions are no longer adequate to the task. They leave things out. The NCI actually agreed with us. They put together some guidelines, they published them in Nature, they planned a big press conference to discuss this. Everybody remembers that press conference, right? Problem is it was never held. Why not? Well, the paper was released under embargo to a bunch of reporters who called the authors at the NCI, who to a person said exactly the same thing. The government is on shutdown, we are on furlough, we cannot talk with you or we will lose our jobs. So, but as it happens, the best summary of what I want at the time of publication is not due to the NCI. It's not from me. It's actually from Brian May. Some of you may have heard of Brian May. Anybody ring bells? He's of Queen. What did he write? I want it all. I want it all. And I want it now. At the time of publication, we've reached the point where, guess what? I shouldn't have to take your word for it anymore. You should be prepared to back it up, and I think I am justified in asking for it. Now, as it happens, I'm an optimist. I'm an optimist because I've been doing this type of stuff for 18, 20 years, and I can tell you the data quality and data sharing expectations are much better today than they were 15, 20 years ago. And in particular, some of the things that I have seen emerge in just the past few years are open source tools to make it easier for people to do it right. And indeed, one of the reasons I am in Seattle right now is because for the past 2.5 days, I've been teaching a short course to a bunch of statisticians saying, here's how you do it. And at the end of that course, I will guarantee that every single one of them was running an analysis and getting exactly the same results as their neighbors got. It's not impossible. If you want to learn more, slides are available, feel free, and these are live links. You can go, you can read about them, that's wonderful. If you want to read more, you can read our papers in bioinformatics and in the Annals of Applied Statistics. I know you've already read those journals anyway, that's okay. I will point out that the Annals of Applied Statistics paper is probably an atypical Annals paper in that there's only 3 equations in the paper and they're all wrong. So just pointing that out. But that's where you can learn more. And there is one last thing I have to, or 2 last things actually. The 2 last things. First off, I have to acknowledge that this is not all me. The reverse engineering, particularly of that second case study that I just told you about, Kevin Coombs and I, between the 2 of us, we spend a bit over 2,000 man-hours reverse engineering all that. And that's probably more time than you want to spend on analyses that should have already been done. But the other thing that I have to point out, or I have to acknowledge, is I have to say, thank you for putting up with me for the past hour, because that's what I got, folks. I am at this point happy to answer questions if we have time. So. I am told to please ask folks to use the speakers so that remote people can hear us.
Fantastic work. One of the things you kind of alluded to again and again was prominent journals basically shutting down solid data, refuting papers that have been in. That's certainly been our experience as well, including a recent experience with Nature, who you probably are aware has an actual format for that. And we had I won't go into the details, but it wasn't a data error, but it was an interpretation error that basically invalidated a study that's now well established. Couldn't get it in. Very similar experience. So my question to you is, it seems like that might be another thing that the powers that be, NCI, NIH, and others might take on, because it seems like it's a systematic problem that journals are reluctant to ever admit that anything they've published is wrong. And you're in a prominent position. Have you had conversations with people that actually can talk to journal editors and affect change in that sphere? Is my question.
So I have talked both with journal editors and with people at funding agencies about this issue, in part. And so we don't have a great solution, but one bit of advice that I got from a medical journal editor, which I have since tested out on a few others, was told was pretty good, is the following. If I were put in the position of having to do this again, God forfend, if somebody rejects the note, he suggested, why didn't you call the editor? The thing is, okay, in statistics, which is where I was trained, you didn't do that. You did that, you were basically asking to have your paper sent back because you were interfering the next day. Turns out that for many of these papers where we raised objections, the authors sent them off to the editors sent them off to the initial journal authors for comment, whereupon Devins or Poti called them up and said, oh, these guys at MD Anderson, they're on a vendetta, they're just out to get us, and things like that. So what I would do at this point is call up the editor and say, okay, you've decided not to take the paper. I'll deal with that. Can you give me 5 minutes of your time so that I can tell you, first off, why I think this is a big problem, and also why I'm not a kook? Because that's actually something they are worried about, because they don't think that they are scientifically qualified to judge this, or they don't for most things. And actually, I think this is something that was a bit bizarre to me, which is that if they'd understood the issues that we were raising, I think they are scientifically qualified to judge things. The labels are wrong. And actually, when we sent that report off to the NCI and to Duke about all the things that we actually wrote a cover letter and tried to simplify it as much as we could. And the first paragraph says, guys, the gene labels are wrong, the sample labels are wrong, the drug sensitivity labels are wrong. These are all wrong in ways that could negatively impact patient care. Didn't happen, but so contacting the editors is the best follow-up I have. But yes, it is acknowledged as a problem.
That's great. I mean, it just seems
I will tell you that about a month after all this was crashing down, I got a phone call from the editor of JCO who said, it is not our journal's policies to keep copies of papers we have decided not to publish, but it has come to our attention that objections you raised 3 years ago were likely correct. Would you be willing to resubmit? And we said, okay. So we sent it back in. That was dealing with cisplatin and pemetrexed. And a week and a half later, that paper was retracted. So our objection has never been published and never will be.
I guess the sort of aggressive thing would be, should the media be contacted about those editors? Because there doesn't seem to be any accountability, there. So that would be the very aggressive move. But
it can be. One of the challenges is you have to be prepared to educate the media or whoever it is you're trying to persuade that this really is a basic issue that they can be convinced they understand, because they're feeling very uneasy
Yeah.
in doing that.
And
but you
I
can
think
do it.
I think your advice was really well taken. Thanks.
Okay. Yes.
Again, thank you for both a great talk and for protecting data integrity and scientific integrity more generally. My question is your advice to those of us, a few of us in this room who may not be statisticians.
Okay.
And as we review papers and serve as reviewers and editors, what's the sort of signal to say
Something's weird.
something weird, or try to convince the editor that this needs a biostatistician's review because they are in short supply, and I think editors often are reluctant to send them to statisticians.
One challenge with big data in particular is that the number of differing fields of expertise required for a single paper is often getting pretty large. One warning sign for me is if a big chunk of the paper is associated with a quantitative method, is it clear from the author list that there are at least a few people that have quantitative training? The Nature Medicine paper has no biostatisticians or bioinformaticians on it, and it's introducing a mathematical method. Second thing, as we look through the paper, first off, if I get a review request and I'm asked to review for Nature, I cannot do a deep data dive in the time allotted for review. I can't do it in a week, sorry. But what I can do is I can say, all right, I'm looking at the paper. I know that I want, and it reads plausibly. I know that I'd really like to check the data and code. Okay, in the paper, do they tell me where the data is? Do they give me a link? Do they tell me where the code is? And if I click on it, is there data? Is there code? And at least at a cursory glance, does it appear like the labels make sense? That's the level of review that I think as a reviewer I could perform. And it's also one where as a reviewer, I feel perfectly comfortable in telling the editor, I'm prepared to okay this as soon as the following is supplied. I'll tell you that most of my collaborators, if they get a note from the editor of Nature saying we're prepared to publish your paper as soon as the data is posted, the data will be posted pretty fast. So, but that's the simplest filter I know to get most of the stuff.
What's the role of clinical labs in showing their work?
Sorry, what do I feel
You talked
about?
a lot about the NIH and journals and these sort of things. You know, they can compel you to, they're not going to give you money, they're not going to publish your paper. What do you think is the role now in like reference labs, clinical labs, in showing your work as we get higher and higher like data-rich assays?
Well, if you are expecting your work to have credibility and be adopted by others, if you are not willing to post data so that others can look at it, I, at any rate, will be telling folks at MD Anderson, we will pay attention to them later. And we won't trust it now. It is also something where we have talked, so you're right, the funding agencies only have governance over stuff that they fund. The FDA does have governance over stuff that they regulate, and they can and are prepared to mandate data supply to them, not necessarily to everyone. As came up in a discussion with one of your folks here today, maybe it would be possible for the FDA to suggest that validation data, anonymized, that proves that this works, should be made public. I think that that would be a perfectly fine idea. Now, note, I am not speaking for the FDA, but that's something that I think is a reasonable thing to ask them. As it happens, I'm going out to the FDA this fall, so I'm going to ask them. So.
All right, let's thank our speaker one more time and